A-Level Biology 9700, Paper 5, Variant 2, May June 2025. And in this video, we will discuss the question number 2. And question number 2 is, sponges are immobile aquatic animals that live on rocks and sediment at the bottom of salt water environments. Figure 2.1 shows an example of a sponge growing on the seabed in a marine habitat. There are many different species of sponges. Most species of sponge are sensitive to environmental stress. Human activity causes some environmental stress. So scientists sampled the marine habitats along the length of coastline in Algeciras Bay in southern Spain to study the species diversity of sponges. The scientists selected 12 sampling stations A to AL as shown in figure 2.2. So the scientists noted the land use or human activity along the coast next to each sampling stations. So they were from A to L, and these were the different areas or the sampling stations from where they got the samples. So the sampling stations were chosen to compare the effect of land use or human activity on the species diversity of the sponges in the bay. At each marine sampling station, the scientists placed permanently line transects 50 meter in length on the seabed, photographed all sponges sighted at a distance of 1 meter either side of the transect, sampled each transect for the same length of time, sampled each transect 4 times a year. Scientists standardized some variables. They two other variables that the scientists should standardize in this investigation. So to accurately compare diversity across 12 different uh, stations or the locations as from A to L, uh, there must be some uh, environmental factors or the variables which should be kept constants. So there are different environmental factors which naturally can affect the sponges. So the additional variables to standardize should be number one, the depth of the water from where they got the samples. So we will write because it will be easy to compare depth of water because significantly affects light levels and pressure. So depth of water should be the same, which affects their different sponge species can grow. And the second can be the distance. The same distance. You can see that distance from shore. So this can be the second point. Second variable which should be kept constant. So because the environmental conditions like wave energy, often change as you move further from the coastline. So these two variables, or there are many other variables which should be kept constant. As for example, we can say that the substrate type should be the same because the sponges need a hard surface to attach to. So comparing a rocky seabed to a sandy one would yield different results regardless of human activity. So in this way, uh, these are the which can be written. You can say that. Now the part B of this question is the scientists compared the species diversities of sponges between different sampling stations using an index of diversity known as beta diversity. The higher the beta diversity index, the higher the species diversity at a sampling station. So figure 2.3 shows a graph of the beta diversity index for each sampling station. So uh, here you can see in figure the highest diversity is between 1.6, this is the, so this point, it can be 1.64 or 1.65, you can see that 1.65. So we can use any one of these. So now moving, a simplified formula for beta diversity is shown. Beta diversity is equal to n divided by x minus 1. X, whereas the n is the number of sponges species recorded at a sampling station. So and x the mean number of sponges species across all sampling stations. So a sampling station A, 37 sponges species were recorded, n is equal to 37. So use figure 2.3 and the formula for beta diet calculate the mean number of sponge species across all sampling stations. So here you can see they have given n is equal to 37 and A I have 1.65 and we have to find out the mean value. So according to this formula, we are beta diversity. A means this is the beta diversity. So for beta diversity in this formula, we will put the values as beta diversity is 1.65 is equal to 37 divided by x bar minus 1. So if we cross multiply these values, so it will be 1.65 multi x bar minus 1 is equal to 37. So x bar minus 1 will be equal to 37 over 1.65. So x bar is equal to minus 1 is equal to 37 over, if we make use of the calculator, then we can have, we will divide the 37 with the, so it is equal to 22.4 and x bar will be 22.4 plus 1 is equal to 23.4. So it will be the answer. So the mean value will be 23.4. So it was the part 2. Now we are moving towards the next part of this question 2. So the next part is a student concluded that the species diversity of sponges was higher in the sampling stations where there was less human activity. So using the information in figure 2.2 and the figure 2.3, evaluate this conclusion. Use the values in figure 2.3 to support your answer. So we will move back towards the question, uh, figure 2.2 and 2.3. You can see the A is natural habitat, B is also natural habitat, and CL is also natural habitat. Other AHA have. So if we watch, we can see in the natural habitat A and B, the highest diversity is there, and L also has the highest diversity. So these 3 points support the answer, that is where the process is the that is species diversity of sponges is higher in the sampling stations where the human activity was less. So it supports the answer, but we can see that the point D point D is housing, and it means where there is housing or the human activity is at its most, you can see, so it will not support the I. So according to this, I'm going to write this answer. So I'm writing the answer. First of all, I will write the evidence which will support the conclusion. So stations with natural habitats, generally shows the highest levels of diversity. Diversity. Another L is also natural habitat, shows about 1.43, but less than A and B. So both of these natural habitat shows. Whereas the uh, station G, which is thermal power station, have showing the uh, lowest diversity. Station G and station I shows the lowest index as 0.71 and 0.81 respectively. This both of the thermal power station and station or shipbuilding you can see. So we would go with the evidence which are not significant. They are not going with the question. So they values shows against this. So station D, this is the point one, and this station D being a human developed area. Human developed area, but shows highest diversity index 1.59, which is higher than stationary. Well, station J and K shows a significant upward trend. Upward trend so with students partially concluded, partially quoted quoted but overwhelmed simplified as some areas still maintain high diversity. Developed areas, which are developed areas. So partially go with the conclusion. Now the part two of this question is Use the information in figure 2.2 and figure 2.3. State two other conclusions about the species diversity of sponges in Algeciras Bay. Use the values in figure 2.3 to support your answer. So, industrial activity has a greater negative impact on diversity than tourism. So, this can be the one conclusion. And if we support over this uh, conclusion, we can say that the station G and station I have the lowest beta diversity index in the study at 0.71 and 0.81 respectively. So tourism evidence station C and station K have higher diversity index of 0.97 and 1.33 respectively. These activities are less detrimental to sponge species. And if we go with the second location within the bay influences diversity levels. So that would not uniform, it appears to be higher at the outer edges of the bay compared to the inner, more sheltered northern sections. So outer bay stations. So in this way, I would go with these two conclusions. The first conclusion is that industrial has a greater negative impact on diversity than tourism. Industrial stations G and I have lowest beta diversity. Beta diversity index in the study at 0.71 and 0.81 respectively. Station C and K shows tourist activities have higher diversity indices of 0.93 and 1.33 respectively. And second is location within the bay. Bay influences diversity level. Levels. So the diversity is not uniform. Is not uniform and appears to be higher and appears to be higher at the outer edges of the bay compared to the outer bay is A and L, and inner bay stations are G, H, and I. Show a significant dip in diversity with ranging from values ranging from 0.71 to 0.94. So it was all about paper 5, variant 2, May June 2025. So please like, share, and subscribe my channel. Thanks for watching.